I welcome you all to this 11th Professor K.R. Laumas Oration organized by Department of Reproductive Biology, AIMS, New Delhi. We have with us our Honorable Director and Honorable Dean with us. I request Professor and Head of Department of Reproductive Biology to introduce the oration and the orator, please.
Very good afternoon, Director Sir, Dean Madam, dear colleague and students. This oration is in the memory of late Professor Kesho Laumas, who is a pioneer in reproductive sciences and also first professor and head of the Department of Reproductive Biology, AIMS. And this is the 11th oration. Professor K.R. Laumas, he is a PhD from India, and later on he did MD from Mexico and residency in psychiatry from USA. He was born on 1933 in India and died on 1995 in USA. Department was created by Reproductive Biology, Ford Foundation and Government of India in 1963 as a reproductive research unit and full department since 1970. And he was the first appointee as a research officer of the department. Later on, he became professor and head of the department. He, among notable works, he conducted 5th International Congress on Hormone Steroid in 1978. And he was that time organizing secretary. He was president of Endocrine Society of India, trained in reproductive endocrinology in NIH, Harvard Medical School, and he did residency in Columbia University Medical Center, New York. Professor K.R. Laumas oration is given. We invite usually an intellectual, either scientist or clinician working on reproductive sciences to deliver the oration. Previous orators, there were 10 previous orators. First one was Dr. A.K. Mukhopadhyay from Germany, and last one was Dr. P. Padmanabhan from USA. And in between, there are many, including Dr. G.P. Talwar and Padma Shri Indira Hinduja, and Dr. B.N. Chakraborty, and A.J. Rao from ISC Bangalore. This year oration is awarded to Dr. Muhammad Abdulkar Akbarsha. He is a PhD scientist working on male reproduction and toxicology. He is at present honorary research coordinator, National College, Tiruchirappalli, Tamil Nadu. He is also editor-in-chief, Journal Endocrinology and Reproduction, and General Secretary of Society for Alternatives to Animal Experiments. He is a PhD in zoology, endocrinology from Madras University in 1980, professor of zoology, Trichy, then deputy controller of exam, research and life science, BDU, reader, director, professor, all from BDU Trichy, coordinator, School of Life Science, dean, faculty of science, BDU Trichy, member of standing committee of and Senate, BDU Trichy. This year also he received Lifetime Achievement Award of ISSRF 2022. He guided 22 PhD students, editorial board member of various journals, including Altex. He has more than 250 publications in index journal. Maximum impact factor journal is Altex, 6.18 impact factor, edited 2 books. His research area is identification of lead molecules from among new and novel metal complexes, biopolymers, quantum dots, animal products, and phytochemicals as cytotoxic agents for drug discovery for cancer, particularly breast and cervical. Then to bring up novel culture system for 3D culture of cells for assessment of toxicity of chemicals entitled as disease models for drug discovery. His interest in andrology, toxicology, endocrinology, and biomarker and pheromone. This time, oration talk is on milestone in the journey of an andrologist in pursuit of exciting discoveries. Now, next 30 to 40 minutes is for Dr. Akbarsha. Sir, I will request you please make your talk within 40 minutes. Thank you.
Okay.
Dr. Akbarsha.
Respected Director of AIMS, the Dean, Dr. Ashutosh Halder, the Head of the Department of Reproductive Biology, Dr. Chaturvedi, my dear colleagues from the other departments and reproductive biology department of AIMS. Good evening. And before I commence, I apologize for the problem in the delayed start. It is because of difficulty in bringing up the full screen of my presentation. Yesterday we did at rehearsal, it was working out all right, but today it is a problem. I'm sorry about it. Well, at the outset, I would like to place on record my appreciation and admiration to the selection committee of Dr. K.R. Laumas Memorial Oration of AIMS, New Delhi, for choosing me to the 11th speaker of this oration, and that is happening today. I'm really sorry that I'm not able to be really present for the meeting in person. I would have loved doing that, but for the problem of this pandemic, that it's a third phase, and particularly it is becoming very difficult for people to move about in Tamil Nadu. We have lockdown on weekends and late evenings. So I'm really sorry about it. But then I shall try to make it as good as possible through the online mode. And of course, I am aware that there are people who are offline also participating in this meeting. So I am really very happy to be associated with it, Dr. K.R. Laumas Memorial Oration. Well, I had suggested 3-4 topics to Dr. Haldar, and he preferred this topic, milestones in the journey of an andrologist in pursuit of exciting discoveries. And I'm really grateful to him for choosing this topic, because when I was preparing the PowerPoint, I found it really very exciting. Really thrilled to be reminiscing my good old days engaged in research in andrology. Well, I have chosen this topic to make a comparison. Well, in my basic theme of research has been andrology, reproductive biology, and toxicology. And the opening slide is a little bit of a reflection of what I am going to say. I have 2 pictures, one on the left and on the right, and you have the section of the seminiferous epithelium. You would see the seminiferous epithelium with the cells in the spermatogenesis in different grades, starting from the spermatogonium to elongated spermatids, spermatozoa which released to be ready to be released. And on the other right, this is also a seminiferous tubule, but in the case of a particular toxic inflictment treatment of an animal model, the mouse, with a toxic chemical, and you could see that instead of spermatozoa to be produced, you are seeing large giant cells, or what you call as the symplas. So looking at this spermatogenesis per se, and I am also giving on the opening screen the spermatozoa. What you see here on the left are the mouse spermatozoa, as you would collect them from the epididymis or from an ejaculate, a beautiful spatulate head with sharp tip and the long tail. And in certain exposures, the animals produce sperm of not this kind, but sperm very different, and we call them as spermatozoa that are very abnormal. So the andrology has been like trying to understand the normal thing that is happening in the time of spermatogenesis, maturation, and fertilization, and also at times when there something untoward happens to any one of the mechanisms of spermatogenesis, maturation, and fertilization. Well, something different happens. So, having given this introduction, I move on to the PowerPoint. Well, my journey into reproductive biology or andrology started with my post-graduation, which was 1968 and 70 at Government Arts College, Coimbatore, when there was the only college in the whole country which was offering MSc with endocrinology as the optional subject. So I was at that age of 2021, I was exposed to endocrinology at its depth. And at that time, in January 1970, I had been to the University of Madras when I happened to meet this person, Dr. P. Govindarajulu, who was just joined at the University of Madras. He was the founder, Department of Endocrinology, Dr. A.L.M. Postgraduate Institute of Basic Medical Sciences, University of Madras. Like Dr. Laumas, who established the first ever Department of Reproductive Biology in the country, Professor Govindarajulu was the one to have started a Department of Endocrinology in the Basic Sciences University, and he was a terrific motivator. My meeting with him further strengthened my interest in endocrinology and reproductive biology. So that was the turning point in my life. So I had chosen at that time, even before post-graduation, that I should continue to do something in endocrinology and reproductive biology. Well, after post-graduation in June 1970, I was chosen to be a faculty in the Jamal Muhammad College in Tiruchirappalli in Tamil Nadu. And that is where I had a very humble beginning as a teacher and as a scientist. And from there on, I have been working in various aspects of endocrinology and reproductive biology for well over 50 years. And I'm so happy that I could reach great heights to the extent of having been invited to be the speaker at this meeting. So at Jamal Muhammad College, when I joined in 1970, we had a professor by name Professor V. Hari Rao. He had just returned from University of Connecticut in USA. He is basically an entomologist and he was starting a research department. So I asked him if I would join him for a PhD. He was readily agreed. And this is me at that age of 1973. So he consented to guide me on a topic in endocrinology, and that too in a model which is very different, and that was the Calotes versicolor. So I started in 1974 under Dr. Hari Rao as a supervisor. I started as a scholar, and I worked on the structure, development, function, and function of the parathyroid gland of the Calotes versicolor. So that was the time Professor J.P. Taplial was choosing this model, the reptilian model, Calotes versicolor at Banaras Hindu University. A lot of research was going on, like Chandana Halder, Woman Be Woman, and many others were conducting studies, research on Calotes versicolor. So I conducted research on this animal to study the parathyroid gland of Calotes. And there was a time after my thesis, that was in 1980, I was slowly getting into aspects of reproductive biology. And it was fascinating that in 1988, we had the Physiology of Reproduction by Ernest Nobel and Neil. It was published by the Raven Press, and that was a beautiful review of all that was happening until that time with regard to physiology of reproduction. And that was a terrific attraction to me to move further into aspects of reproductive biology. And then in 1989, I had invitation to move from Jamal Muhammad College to Bharathidasan University as an associate professor, and that happened in 1989. And that gave me much more opportunity to be going deeper into research on topics that would be really interesting and exciting to publish good papers and produce good students. So that was in 1989. And another turning point that came up in 1998, I had started working on reproductive biology. And in 1998, I attended a conference, Borden Conference on Epididymal, that happened in Sydney in Australia in 1998. And the people who wrote the book, The Physiology of Reproduction in 1988, I had the pleasant meeting with many of those people who were contributing to that book, particularly Dr. Dee Crester, who wrote about the testis, Dr. Bernard Rubaire, he wrote about the review of the epididymis, Dr. R.C. Jones, he wrote about the reproductive biology of lower vertebrates, Dr. R.G. Binkrist, Dr. R.C. Jones, Dr. X.A. Hess, and many others. And it was really exciting. And I said, because we talk about milestones, this meeting with these very powerful endocrinologists and reproductive biologists, the meeting was really very inspiring. And I took a firm decision that this is my topic, reproductive biology, with special emphasis on epididymis, looking at sperm maturation and ejaculation. So that was the milestones. So from around that time, my lab has been engaged in research on aspects of male reproductive toxicology and male contraception. Well, I am the person today in the whole of the country, the most authoritative, the most powerful in respect of microtechnique and electron microscopy. So I was adapting histology, histochemistry, histopathology, and a little bit of biochemistry and immunohistochemistry, and a great lot of transmission electron microscopy and spermatology as the tools in my research. And I was targeting spermatogenesis, spermiogenesis, and Leydig cell biology, looking at the testis, and at post-testicular sperm processing, with special reference to things happening in the efferent ducts, epididymis, vas difference, ampulla ductus differentia. Thus, the aim was to unravel the several mysteries of male reproductive biology or andrology, and to apply the knowledge in understanding the mechanistic perspectives of male infertility and subfertility. That was my emphasis, not to really look at what really happens normally. That was the time when a lot of things were spoken about infertility among men. 10 to 16% of men were reported to be infertile, and that was very, very emotionally spoken about. So I was trying to look at the causes underlying the inflictment of male infertility. So I was interested in finding aspects of male infertility and subfertility. And also there was a time when people were trying to look at male contraceptives and look at epididymis as a target for male contraceptives. So this is how my research went about. I was in animal models in my study. I used Easter rat, Swiss mouse, and Mohd monkey. And I tested pesticides, namely phosphimidone, dichlorvos, and malathion, and dietary toxin, aflatoxin B1. Each one has a history as to why I choose this. Environmental or occupational heavy metal contaminant, which is chromium. I had a collaboration with Dr. Michael Aruldas from University of Madras to tackle this issue, particularly when people in the leather industry and chrome plating were exposed to chromium, and they were reporting to be, many of them were reported to be infertile. So we were trying to look at the same thing in the monkey model. Then medicinal plant extract, we tried Andrographis paniculata, which is the medicinal plant, which is now greatly used in the context of chikungunya and dengue. That's the medicinal plant. Phytotherapeutics, pure compounds coming, Vincarosia vincristine, piperine from Piper nigrum, plumbagin from Plumbago zelanica, andrographylate from Andrographis paniculata, ursolic acid from Ocimum sanctum, quasin and neoquasin from Quassia amara. So these were my substances which I was testing in these animal models. And incidentally, I also had interest to look at the human case study. I had collaboration with the Dr. T. Anand Kumar at Hope Infertility Clinic in Bangalore, and he was providing me with sperm samples coming from patients reporting to infertility clinics. So that is where I went about. Well, this is initially I speak a little about what I did with respect to testis. And what you see here are two pictures. One on the left is the hemotoxin used in stained section of the seminiferous epithelium. And I spoke at the very opening slide, starting from the spermatogonia, you get the tiers of cells ending up as the spermatozoa or elongated spermatids. This is TBO stained section, much more illustrative. And you look at the spermatozoa, what normally sperm would look like. This is the human sperm. This is not my own picture. This is an adapted picture. This is the spermatozoa. These are the spermatozoa of the rat, the normal rat sperm. Well, when that be the case, I had to look at, for some obvious reason, aberrant spermatogenesis resulting in multinucleate giant cells, otherwise called as symplast. Russell Jones was using this term symplast long back in 1994 and adapted that terminology. And another was multiple micronucleate giant cells. And it was a serendipitous discovery in the mouse, because these mouse were used in an experiment, and I was feeding them with the pelleted feed. And I found very aberrant things happening in the testis and epididymis. And I was advised by my peers that this is not really what is happening in response to what I was testing in these animals, but probably there is something in the contaminating the feed the animal was consuming, and we tested it for aflatoxin, and we found that the feed was contaminated with aflatoxin. So that became the opening scene for me to enter into testing of aflatoxin B1 in the mouse model, and many fascinating discoveries were made. And what you see here are the seminiferous tubules of the mice who were treated with aflatoxin B1. And this is not accidental exposure, but was deliberately an experiment was conducted. And as you would see, you don't see anything semblance of a spermatogenesis in a normal seminiferous tubule. And you have a magnified view of a seminiferous tubule, and you will see, don't see even spermatids, you don't see spermatozoa or elongated spermatids, but you see a large football-like cells, and we call it this multinucleate giant cells. So remember, aflatoxin B1 on exposure to mice, the mice don't end up in spermatozoa, or at least not as many spermatozoa as normally are produced during in spermatogenesis. At least 40 to 50% of the spermatozoa were not really produced, and in place we are getting such the aberrant cells. And what you see here are those cells which were very unusual to see in the seminiferous tubule. I call them multinucleate giant cells. So here is a large cell with many nuclei in it, like roughly about 16 nuclei in each of these cells, and I call it multinucleate giant cell. This is in the mouse model when I was testing aflatoxin B1 for toxicity. So I got interested, not to end up with just reporting that the aflatoxin treatment brings about generation of multinucleate giant cells and not spermatozoa, the normal way the mouse would produce. Then I got interested in finding out as to what exactly would be the mechanism underlying the aflatoxin-induced aberrant spermatogenesis, ending up not in spermatozoa, but in unusual cells of this kind, the multinucleate giant cells. So we went to transmission electron microscopy to find out really what happens during the time when these cells are produced. And what we found was that we could see binucleate giant cells, tetranucleate giant cells, and and number of nuclei more than this, from 8 to 16 nuclei were seen. And as you would see here, it is actually formed from either 4 nuclei or 4 cells, or 8 cells, or 16 cells, with their nuclei all brought together in one common mass. We call it the multinucleate giant cells. So we try to look at exactly what would be the mechanism underlying the formation of such giant cells. And we found that in the case of, this is a case where you get 4 round spermatids. Each one is the 4 round spermatid, here 4 round spermatids. But what has happened is that these down spermatids are not separated from each other. What really is seen is that the cytoplasmic bridge, it is physiological, it is normal. The cytoplasmic bridges are bridges that connect the round spermatids until the spermatids are fully elongated as spermatozoa. When the spermatozoa separate out, the bridge collapses. It should collapse. But in this case, because of the aflatoxin treatment, these round spermatids were not separating out. And instead, as you would see, the cytoplasmic bridge connecting the spermatids are becoming wider, widened up. And as a result of which, you could see that cytoplasm from these 3 cells has been pushed into one of the other cells, the 4th cell. And you could see, unlike this, here you have smaller cells, and one is a larger cell. The larger cell has more cytoplasm. The smaller cells have little cytoplasm. So the cytoplasmic bridge is still open. And we could further trace it. This is not a sequential. You don't get anything to really look at and expect it to be happening like this, but we are only putting the photographs together to tell a story. And as you would see here, you can get a cell which is cytoplasm rich cell and a cell which is cytoplasm poor cell. The bridge is still open. So and through this bridge, the cytoplasm is pushed into the larger cell. And you can see that very fascinating, fantastic, as you would see. Not only the cytoplasm, but you have the cytoplasm rich cell and cytoplasm poor cell. And through the bridge, you could see even the thimble-shaped nucleus moving from one cell into another cell, as a result of which you have now the plasma membrane of the 4 cells remaining continuous with the nuclei arranged around a central point. And you could see that the Golgi apparatus, which was to develop into the acrosome, is not differentiating to that point. And you can see that it is a spherical mass with 4 nuclei and no acrosome to develop. And further to trace that, we had many other links, but for convenience, I have chosen this particular material. As you would see here, once this has happened, the cells are not ending up as spermatozoa. When the spermatozoa are not produced, and then naturally, well, the spermatogenesis is aborted, and you can see that the nucleus is not ending up as the nucleus of the sperm. So the nucleus undergoes a kind of the whole cell is going to die. And you can see here. What see in the next stage was that in these cells, the cell was not a spermatozoa, but they succumb to death by apoptosis, as you would see the crescentic nucleus marginalized to chromatin, and you can see that is how the multinucleate giant cell is originated, and it ends up apoptosis. And very fascinating, interesting thing is that these are cells would ultimately arrive at the epididymis in the same manner as would normal sperm arrive, but with some difference in that the cells are in the process of death and disintegration through apoptosis. So so this part of the study was reported. Aflatoxin treatment brings about generation of multinucleate giant spermatids at symplas through opening of cytoplasmic bridges, light and transmission electron microscopic study in the Swiss mouse. And that was the first ever study to be reporting this kind of aberrant spermatogenesis ending up in giant cells through the opening up of the cytoplasmic bridges. And to date, it remains the only publication which has been reporting such a fantastic discovery. So having done this, we had another observation made in the same mice, but this time in the epididymis. What you see here is the lumen of the epididymis with its epithelium. Rather than spermatozoa, you are getting large cells, but these cells are not the same as the one which I was showing earlier. You're having here again, it is a giant cell, and you get small dot-like material, which are not really nuclei. We got again fascinated. What is really happening here? It's again mouse model treated with aflatoxin B1, and the multinucleate giant cells are produced. Of course, that's a different observation, but over and above that, we could see these large cells with small chromatin bodies, and we started investigating further on that. And we found that, so this is HND stain section, and this is these are TB stain sections. So what you see here are not nuclei. You get chromatin bodies, or you might call them as chromatoid bodies. And this is a different manifestation from what you have seen earlier. You have a picture here. This is the cell this is again a giant cell, but what you see here are not the nuclei. Whereas this is the earlier one which I showed, discussed earlier in the previous paper, large multinuclear giant cells. So here what you are getting is a little different. So we try to find out what exactly is happening there. And we found that, cutting short, we found that this is a case where, because of the aflatoxin treatment, this is not actually a multinucleate giant cell being produced, but something is happening to the spindle apparatus, as a result of which you can see that the chromosomes are aligned in the equatorial plate as pairs, but beyond that, the development is not progressing further. The spindle apparatus is affected, and as a result of which, you can see that the paired chromosomes remain as chromatoid bodies, and the chromosomes are not going to move to the pole. The reason being, you are not getting the spindle apparatus. So the aflatoxin is, in addition to being a genotoxic material, is also a spindle poison, and it's disrupting the spindle apparatus. As a result of the paired chromosomes don't separate out, and they become they're condensed into chromatoid bodies. We call these cells as multiple micronucleate giant cells. Please compare this with the earlier statement, multinucleate giant cells. There they are nuclei. Here they are not nuclei, but they are actually pairs of chromosomes. You can see that here you get many such cells were produced. And here again, like in the earlier case where multinucleate giant cells were succumbing to apoptosis, here we found a similar mechanism, but it's not exactly apoptosis, but something comparable to apoptosis. So you could see that in the testis itself, the chromatoid bodies undergo a kind of a disintegration, and you could see such a thing happening in the testis and later in the epididymis as well. So this was again, we reported it, induction of meiotic micronuclei in spermatocytes in vivo by aflatoxin B1, light and transmission microscopic study in Swiss mouse. So the same treatment, aflatoxin B1, serendipitously discovered in mice which accidentally fed on aflatoxin contaminated feed. Later, we carried out very clearly directed aflatoxin at 20 μg per kg body weight, and it was treated to mice, and we found multinucleate giant cells and multiple micronucleate giant cells, telling us that the aflatoxin, whether it be mouse or human or chicken or buffalo, when these are, including man, happen to be through our feed, like bread mold, we happen to take the bread mold, we can have our spermatogenesis being hampered to the exposure to the aflatoxin. So we had wonderful science coming up, and we could publish it. And having done something about the testis, I take you to the male reproductive tract, where under such circumstances of toxic inflictment, we try to see what happens to the epididymis. So male reproductive tract. Well, I'm not going into the details of the epididymis. You get the anatomical segments of the epididymis, the initial segment, intermediate zone, caput, carpus, and carda. And the mouse and human epididymis compared. The human epididymis doesn't have the intermediate zone. In mouse, it has an intermediate zone, and you get different segments. And this is the first ever uncoiled ductus epididymoides, that is called as the D-graft thread, that was done in the 18th century. To explicitly say that the entire epididymis is formed of single, long, convoluted, contracted ducts originating from the testis. It goes on like this, and it tells about it is a single duct, single long duct with a single layer of epithelium with several types of cells like principal cell, basal cell, halo cell, clear cell, and other cell types. Okay, so I had the fascinating experience of dealing with this heterogeneity of the cell types and the heterologous or the heterogeneous differentiation of the single duct into the initial segment, intermediate zone, caput, carpus, and carda, continuing as the duct is different. So the epididymis is associated with absorption of material, fluid, and ions. It concerns the secretion of proteins and other molecules. All concerned with metabolism of steroids and intermediate metabolism. It is concerned with the spermiophagy. It is phagocytosis of defective sperm. And these are all essentially happening towards one single goal of the post-testicular physiological maturation of sperm. And therefore, this is one of the most complex of the organs and most organ with the most complex function, ultimately rendering the sperm become motile and fertilizable. So these two things happen to the sperm during the sojourn or transit of sperm along the ductus epididymalis. And these are all the changes that happen to the sperm during the epididymal transit. And the major changes you get, particularly with reference to proteins, and this protein alteration renders the sperm undergo maturation, right? So this is another adapted picture, a cartoon, to tell that from the testis, when the sperm arrive into the epididymis, they are not motile and they're straight. And as you would see, there will be material secreted from the principal cells, and these proteins and the other molecules interact with the spermatozoa to slowly acquire the aspects of motility. And when the spermatozoa arrive at the carda, you could see these sperm are capable of becoming motile. So the whole thing happens when they transit along the epididymis. Well, this is the complexity of the epididymis, which I was dealing with for well over 30 years. So I could, in one section, I could easily identify what is the initial segment, what is the intermediate zone, what is the caput, what is the corpus, and how would the carda look, the duct of the regions, right? So that was fascinating. So this is another picture to say that. So you get different types of cell types, because I was concerned a great lot about the cell types, and I discovered at least 2 new cell types during my journey with the epididymis. So you have principal cell, narrow cell, apical cell, basal cell, clear cell, and halo cells. Well, these are the cell types which people like Bernard Roubaire and Hermo had already identified and described well over like 10-15 years. And we took off from there. By my interaction and association with those experts, I could not only look at what happens to each one of these cells due to toxic manifestations, but also to see if there are more or additional cell types. Well, you could see, these are my own pictures, and you could see these are the principal cell. This is stereotype section, and you have the apical cell, principal cell with the narrow cell, and you have the basal cells, have the clear cell here. Well, each cell has its own functional significance. They are not similar, not only morphologically, cytologically, ultrastructurally, but in terms of function also, these cell types are very different. Principal cell are the most predominant, 60 to 70% of the cells are principal cells. They are the ones which secrete the different kinds of proteins which interact with the sperm and render the sperm motile. So having said about it, this is one of the finest of pictures which I ever take. I used to say that the principal cell of the epididymal epithelium is the one with the most complex Golgi apparatus. And I used to illustrate it with this picture. What you see here is the most complex Golgi apparatus that I have ever come across in the principal cell. Well, the very special organization of the different cell types. Well, with my experience, you show me any cell type, I'll be able to very clearly identify whether it is the apical cell, or it is the basal cell, or it's the intraepithelial lymphocyte, or narrow cell. Well, I had the exciting time looking at these cell types. These are the clear cells, the basal cell, the intraepithelial lymphocytes, which are either lymphocytes or macrophages. Very, very, very interesting. And we found that, having said about it, we found in the animals which were exposed to these toxicants, as much as there were very serious changes taking place in the testis with regard to spermatogenesis, we found many other things, similar things happening in the epididymis as well. And that is what I spend more time with the epididymis than with the testis and the rest of the male reproductive system. What you see here is in the case of toxic inflictment, you can see that the entire epithelium in the epididymis is hypertrophied and it has undergone hyperplasia. So you don't have a lumen at all. And this is interesting because if the testis is normally in spermatogenesis and the spermatozoa arrive at the epididymis, where do they go? After all, the whole lumen is obliterated. We have interesting answer to come. Another, you remember the multinucleate giant cells and multiple micronucleate giant cells produced in the context of aflatoxin treatment. You could see these giant cells produced there in the testis, and in the same manner as would spermatozoa arrive at the epididymis, these giant cells, the multinucleate giant cells and the multiple micronucleate giant cells, they also happen to arrive at the epididymis. So you have now two contexts. One is that due to aberrant androgenesis, there is hypertrophy and hyperplasia, as a result of which the lumen is almost getting obliterated. So there is no place for the sperm to be there and be moving on to the posterior parts of the reproductive tract. Or alternatively, the whole lumen gets obstructed with these giant cells, and therefore you don't have much sperm, hardly any sperm here. Most of the cells are the aberrant cells in spermatogenesis. So we were trying to look at what is the response of epididymis when there is an obstruction and when there is an obliteration. So you can go through that. And one of the things we noticed was that we could see many macrophages, luminal macrophages in these epididymal ducts, and we could see that these macrophages were phagocytosing the cells which are abnormal spermatozoa or the aberrant cells. So you can see clearly macrophage is phagocytosing sperm which is defective, and very unusually we got evidence to the effect that when there is an obstruction, when the sperm won't be able to move further down to the rest of the epididymis or into the vas deferens, very unusually we could see that they were finding their access into the cytoplasm of the principal cell. What you see here are the microvilli of the principal cell, and you could see the sperm getting access into a cell. I don't know why and how. I mean, why we know, but how the spermatozoa could make it into the lumen, into the cell is not very clearly understood, but we have very clear evidence. See the next pictures, you could see that definitely really these spermatozoa could make it into the cytoplasm of the principal cells. And we know that you get a bloody epididymis barrier, and therefore the epididymal epithelium doesn't permit these cells to be, spermatogenic cells to be accessing into the cytoplasm. Otherwise, it would produce autoimmune responses. But still, in this unusual situation of obstruction or obliteration, you could see that the spermatozoa could make access into the cytoplasm of the principal cells, where they could be entangled, they're closeted in vacuoles, as you see here, and you could see the spermatozoa undergoing a kind of disintegration. And further to that, the spermatozoa, when they happen to access the epithelium from the lumen is filled with not sperm, but more of those giant cells, which I was explaining earlier. And you could see the sperm are going into the epithelium. Very unusual, very unusual. You could see that the sperm could penetrate or permeate the epithelium, the whole extent of the height of the epididymis, the epithelium. But interestingly, there is the point beyond which the spermatozoa are not able to make access into. At the point the question arises, okay, obstruction, therefore spermatozoa, like in a leaky tube, the spermatozoa happen to access into the epithelium. And inside the epithelium, they are able to be present in small vacuoles and get disintegrated. And they rarely make a move up to the basal region, but it doesn't go. The spermatozoa doesn't pass beyond the basal cell. So the basal cell has a built-in mechanism as to a deterrent for such spermatozoa, which accidentally or intentionally happen to access the epithelium. So we could illustrate, we could show that under unusual circumstances, the spermatozoa could access into the epithelium, into the cytoplasm of the principal cell to a certain extent of going up to the basal region, but it does not extravasate. It doesn't, the spermatozoa do not move to the other side of the tubule, unless it would be like an autoimmune response. So what really happens then? The next phase of it, we could say this is all not very intentionally, very clearly directed study. In different contexts, we used to be like a cinematographer, we used to be clicking on, going on, capturing pictures, and try to put them together into well-knit stories. That is what I'm trying to do to you now. So the spermatozoa, when they arrive into the loop in the epithelium, then we could find that you could form into, in the epithelium, you could form into big vacuoles, as you see here. And these are the vacuoles where the spermatozoa, which accidentally access to the epithelium, they would be entangled and closeted, and this be reported. And further to that, we found this is the new cell type which we explained, which we reported. This is what we call as Pain-Vaculated epithelial cell. This is TBO-stained semi-thin section. And we found it initially to be a vacuole, a V, that's what we labeled. And this vacuole is one where you could see either a sperm or a product of disintegration of the sperm, which would accidentally access, because you could see the spermatozoa in the lumen, and this is the epithelium. And in the epithelium, you could find a vacuole. But something attracted us to the effect that, well, the vacuole is lined by a layer which is not like the layer of a vacuole. You can see here, this is the content, the product of disintegration of the sperm. You can see the sperm in the lumen, and you have the boundary, the lining of the vacuole, which is a little different from anything else. So we went further, we tried the transmission electron microscopy, and we found that, well, this is a vacuole which is surrounded or immediately lined by a different cell. What you see here are the principal cell. The principal cell, it overgrows this vacuolated region, and it has its own boundary, its own cytoplasm, its own endoplasmic reticulum, its own membrane, which is different from the membrane endoplasmic reticulum of the principal cell. So you have, we at this time, we called it the pale vacuolated epithelial cell. And incidentally, subsequent to our publication, there has been a group of scientists from Japan who adapted this terminology, and they have also found similar cells. This is a large vacuolated cell, which is, it is a cell different from the principal cell, basal cell, narrow cell, the intraepithelial lymphocyte. This is a very special cell with the animal, the epididymis is able to bring in, establish in the context of the disintegration of the spermatozoa, which accidentally or intentionally happen to be present in the epithelium of the epididymis. So we reported it, pale vacuolated epithelial cells in epididymis of aflatoxin treated mice. Here again, this is in the aflatoxin. Well, we had, I now take you to another in our milestone. I had a very interesting collaboration with Dr. M. Michael Aruddas from Department of Endocrinology, PG Institute of Basic Medical Sciences, University of Madras. So he had done a lot of experiments to look at the toxicity of the chromium, from leather industry, from tanning industry, from chrome plating industry, could affect the human working elements, and therefore we used to do it. I mean, he wanted to look at what happens to a closer model to human, and that is the bonnet monkey. And we got a lot of discoveries using the monkey model. Here, because I spoke about the pale-Vaculated epithelial cell in the earlier illustration, we had a similar thing occurring in the epididymal epithelium. But as you would see here, in the pale-Vaculated epithelial cell, it is the whole one single cell where it is. I'll just go back to that. You see that the whole entire lumen is lined by one cell. Therefore, this is one single pale-Vaculated epithelial cell. Whereas in this case, what we found was we could see junctional complexes between separate cells. So what you see here is the big vacuole, but this is not unicellular. It is multicellular, as you see here in the light microscope and in the electron micrograph. You could see these are all the junctional points. We could see the junctional cellular junctions, and therefore these are vacuoles that are produced by being wrapped up in 4, 5, or a dozen very special cells. So we wanted to, we are at a loss to explain what exactly it is. So we called it micro canals. Okay, we have come across, like, remember in the case of vasectomy in 20-30 years back, vasectomies in some cases failed. The reason being that in the vasectomy cases, there is patenting microcanals developing between the two cut ends, as a result of which the sperm would be able to move from one cut end to the other cut end, causing a failure of the vasectomy. That was reported. Okay, the scientists, particularly like R.G. Priest and others. And here we have almost a similar thing. It's like a patenting microcanal where spermatozoa would be enlarged if they happen to come in. You can see here. See, this is multicellular. This is the whole lumen is multicellular, single lumen surrounded by. So we call that patenting microcanal, where the spermatozoa. I mean, these are all, like, you don't just ask for it and get a picture like this. These were pictures, photographs, which we were collecting, again, hours and hours and hours of sitting with a microscope, and with a kind of a vision that we are going to find something new, which is new to science. So we went on looking for more and more and more of complexities and newer things to report. And we are really been blessed that our keeping alert, our vigilance could lead us to finding more and more of newer material in science. So from pale-Vaculated epithelial cell, we could report the development of obstruction. Obstruction is there in the same manner as per protoxin B1, but here it is chromium-treated animal. Here again, there is obstruction produced due to giant cells and defective spermatozoa that obstruct the tip at the lumen. So the sperm happen to make access into the epithelium, and we could see the patenting microcanal, which is again in the experimental model. We were the first to report it. Okay, so microcanalization in the epididymis to overcome ductal obstruction caused by chronic exposure to chromium. The study in the mature bonnet monkey, Macaca radiata, this appeared in the journal Reproduction, which is later Journal of Reproduction and Fertility. Okay, that's that. So this is about a little bit to tell you about the epididymis. I can speak about epididymis for hours. Well, just to apprise you that we spent a lot of time with the epididymis and made astounding findings and made several new reports in terms of with respect to epididymis, starting from mouse model, rat model, and the monkey model. So having said about it, we had the opportunity to look at, in as much as there were giant cells produced, multinucleate giant cells, multiple micronucleate giant cells, we also found that in these cases of toxic inflictments, spermatozoa were produced, but they were abnormal. So I would like to spend a few minutes how these spermatozoa, abnormal spermatozoa, abnormal sperm morphologies, and the mechanism by which such sperm are dealt with. This is where again we have made a great lot of original discovery and made reports. So what you see here is an assemblage from different contexts. The spermatozoa, this is a acrylic orange ethylene bromide stained sperm, which are abnormal morphologies. And you have now, I'm not describing, you can just look at it. This is kind of a globospermia with the highly coiled tail. Well, that's how it happened. And one thing fascinating was that we found quite many spermatozoa in such cases, particularly in the context of quasine and neoquasine treatment, and in the context of piperine treatment. We are finding that this is in the rat model. We are finding that the outer dense fibrils of the spermatozoa were undergoing a kind of extrusion. And as you can see, it's beautiful to look at. You should have the patience. You should be really amazed to look at spermatozoa with such morphologies. And very interestingly, not only at the principal piece, you could see such things happen in the terminal piece also, and you would see the ODFs are bulging out from the tail. And to prove that they are only the ODFs, we have taken electron micrographs, and we could see that you could see the sperm membrane, and you can see that the ODFs are incomplete. You can see half of the tail has no ODFs. What has happened is that ahead of this point, the ODFs have extruded. Now, the point to explain to you is that the treatment has affected the spermatozoa in respect of its spermiogenesis, in respect of its sperm maturation, the result of which the sperm membrane becomes very weak, and as a result of this, the ODFs happen to be extruded, and these are not spermatozoa which are going to be motile and which would fertilize the sperm, fertilize the ovum. Okay, so we made a separate paper out of this again in reproduction. Spermatotoxic effect of aflatoxin B1 in rat. Extrusion, this is a rat model, not mouse. Extrusion outer dense fibers and associated axonome and microtubule doublets of sperm flagellum. Well, these are some of the illustrations in the aberrant spermatozoa, abnormal spermatozoa, which are produced during these different treatments. And I showed this particular picture very specially for the reason that the editor of the journal was so impressed by the sequential. This is in a normal sperm. The mitochondrial sheet is beautifully present there around the axoneme. And you could see that because of the problem caused due to the toxicity, you could see the mitochondria in disarray and disentangle, and the editor was so excited and fancied by this picture, he took our special permission to put it up in the journal website, okay, in the Reproduction and Fertility. This is a little bit to tell you about that we were dealing with the human sperm also. Similar to what I showed earlier, like in the rat sperm, we also had sperm coming from Dr. T.C. Anandkumar, which I was investigating, and we found a similar thing that was shown in respect of the mitochondrial sheath of the middle piece. And this is again in a human sperm, the ODFs. The entire ODF the ODFs are extruded, and this was not reported anywhere in regard to human sperm. And in the human sperm, one of the most common thing found in infertile men, the men reporting to infertility clinics, we could see that the heads were globose. These are called globosospermia. Not alone that, over and above the globososperma, the head, you could see in each of these heads a vacant space, an empty space, disintegration of the chromatin, sperm chromatin. That is the abnormality that is produced because of the causes of infertility. And you could see again the globospermia problem in the middle piece, particularly affecting the mitochondria. Well, that is Taking you now to the next level of what happens during sperm maturation in the epididymis. Very unusual thing we found was, in some instances, the lumen of the epididymis was abounding with sperm, which were not shedding the cytoplasmic droplet. You know, the spermatozoa carry with them a small drop of extra cytoplasm, the cytoplasmic droplet, which is physiological. Rex Hess was the first to show that this cytoplasmic droplet is rich in aromatase, and it is able to convert androgen into estrogen, very specifically required at that particular part of the male reproductive tract, where the spermatozoa require the estrogen to be present there for a physiological role. And then the time comes when the role is fulfilled, the sperm maturation is taking place, the cytoplasmic droplet is shed. Please remember this. The cytoplasmic droplet should be shed when the sperm has passed the caput, when the sperm passes into the corpus, or at the best the carda. Sperm which arrive at the carda should be devoid of the cytoplasmic droplet. So this is an experimental situation where you could see that the spermatozoa retain the droplet. The droplet is not shed. You could see very special membrane elements present in the cytoplasm of the droplet. The sperm is not expected to be retaining this droplet. So we call it retention of the cytoplasmic droplet. Infertility, one of the causes of infertility in men is the failure to shed the cytoplasmic droplet. So you could see here, this is the carda, where you could see a lot of clear cells. And it is here you are getting sperm with the cytoplasmic droplet. You are not, for any technical reason, you cannot expect sperm to be having the cytoplasmic droplet when they have arrived at the carda. But we see that the sperm retain the droplet. So we could do a lot of study. Retention of cytoplasmic droplet by rat carda epididymal spermatozoa after treatment with cytoplasmic and xenobiotic agents. So that's one journal of reproductive fertility. It's a fairly old, but the publication still remains novel and nascent because nobody else has ever tested our findings and nobody else has made newer findings over and above what has been done. So having said this, I take you to almost the fag end of my talk. Little more about the more recent publications, but data were old but published recently for reasons obvious. And what you see here is another subnormal morphology of the sperm. This is called as the DAG defect or the lasso sperm, which is again produced due to either aflatoxin treatment or treatment with Quassia amara extract or quasin and neocausin, which we had being provided by Dr. Ben M.J. Parora from Burki. He is no more, very sad about it. And we did a conducted experiment because quasin and neocausin were substances at that time being standardized as anti-malarial substances. So we had the company also, the company from Germany, they were willing to provide to us the quasin. What Ben Pereira provided was the Quassia amara extract. So we had both the extract and the quasin pure compound, and we found that when this extract or the substance was tested on the mice, most of the sperm were produced in this manner. So you can see the normal sperm is here, and you could see here the DAG defect, or where the whole flagellum takes a curve, turns on itself around the head, and becomes like a whip. Okay, this is called DAG defect sperm or lasso sperm. So we could produce this sperm in large numbers in animals which were treated with Quassia amara extract or quasin. We found it in aflatoxin treated mice also. So this is an illustration which I showed in the first slide. As you would see here, this again the flagellum is recurved on itself, bent on itself in the form of a whip, and it goes back onto the head and leaves out ahead of the head, as you could see here. So we were really fancied and fascinated by looking at this sperm, and therefore, as usual, I don't stop at that, simply reporting what we found. but try to explain as to how in what way, in what mechanism are these changes are taking place. So I asked my student to look at the various pictures he was able to acquire, and as you could see here, ultimately, I mean, no time to explain again. What really happens is that, you remember I told you about detention of the cytoplasmic droplet. This is precisely what happens in this case. The row of spermatozoa, they were not all together. They were like trimmed and put in place to make into a sequence. We do not know this is the right sequence, but to us, this is the right sequence. And as you could see here, the cytoplasmic droplet has swollen to extraordinary size. That's the point. In a normal case, the cytoplasmic droplet would be of this size, but you could see that the droplet is swollen to extraordinary size, and you could particularly see the membrane elements of the cytoplasmic droplet still retained there, and you could the different appearances, as you could see here, the droplet is still retained here. So here we be strained Epithelium spermatozoa control and concentrated mice displaying different morphological manifestations of the spermatozoa, right? So, and very fascinating, interestingly, well, we were initially under the impression that this would something that occur in the epididymis, when the sperm arrived at the epididymis. And accidentally, again, many of these discoveries were accidental. We don't really aim at seeing at these things with any objective focused on that. When we were looking at the testes of these animals, and we could see here that this lasso, the coiled morphology, is occurring in the seminiferous tubule itself. Okay, you can see here. Here is a sperm with you can see here, seminiferous tubule. This is a seminiferous tubule with the carda, with the flagellum coiled upon itself into the lasso morphology. That is what you see here in the epididymis. So this is to show that this is a lasso sperm. This is not a sperm with two axonemes. There's actually an artifact. But really, what happens is that due to the curving of the flagellum, consequent upon the swelling of the cytoplasmic droplet, you could see fusion of the membrane, as a result of which you are getting two axonemes. Actually, this is two. They are axoneme along the length, single axoneme along the length of a tail, but during the coil curve of the flagellum, when you take a section cutting across here, you get a section like this with something like two axonemes. So that was a fantastic discovery we made. So I'm not reading this, and we published it recently. Observations on DAG-like defect of spermatozoa induced by treatment of the phytotherapeutic Quasiamara faucin in the mouse model. It appeared in Andrologia by Wiley, and it was published in 2021, February 21. Well, we were really aghast with the discoveries which we were making. These were not, I tell you again and again, these are not very clearly directed hypothesis-based research. We knew that we were expecting the treatment to produce abnormal outcomes, but we were not really aiming to look at the DAG defect of the lasso sperm. We had it, and you can say that one should have the luck or playing on you. Well, this is the last the science I'm speaking about. I don't know whether any of you would have really seen by making analysis of the seminal plasma, looking at the semen of animal semen or human semen. We had the luck of looking at semen. When we were making observation of semen under microscope, we found a semen with a lot of dense matrix. This is a dense matrix, and in the dense matrix, you could see plenty of spermatozoa. Well, you have also spermatozoa outside the dense matrix. Okay, these are two dense masses, each mass with the spermatozoa, and also sperm with outside the dense matrix. Under careful examination, we found that those spermatozoa, which are entangled in the dense matrix, are abnormal. And those spermatozoa, which are found outside this dense matrix, in the normal seminal plasma, you will get them as all normal. So something new to explain. We have found it. We have found it in ejaculate, and we have found it in a human ejaculate as well. This was, of course, in the mouse, but we found it in the human ejaculate, which Professor TCA was sending us. And we then put up a story like, well, this is a case where we presumed that the dense matrix is a mechanism whereby abnormal spermatozoa are secluded from normal spermatozoa, such that these abnormal spermatozoa could be further acted upon by something present in this dense matrix that would ultimately disintegrate and remove the spermatozoa. So the defective spermatozoa. The normal sperm are totally secluded from these defective spermatozoa because of the dense matrix. So the dense matrix is a mechanism of totally secluding the normal sperm and protecting them from any enzymatic onslaught or free radical onslaught, and seclude the defective spermatozoa and do something to them. I will track these changes. So having seen this, we went to look at what really was happening. This is a case where, okay, this is actually, you can see here, from the epithelium of the epididymis, you could see large protrusions, which are produced like, these are, it means I cut short, like these are ones which are called aposomes. Epididymis is known to produce these aposomes. The principal cells, under certain circumstances, rather than secreting neurocrine proteins, can produce such kind of specific discrete masses of proteins. And these masses could be extruded into the lumen. And you can see these masses amalgamate among themselves, fuse among themselves, and form into a continuous mass. So we went on to track it further, and we found that these aposomes, when they are released into the lumen, we made further observation of these aposomes, and we found that the aposomes had some kind of a very discrete particles in it. This was not reported earlier. Aposomes were known, and what we saw as the discrete particles to be present in the aposomes, we identified them as epididymosomes. You know, epididymosomes are also known. It was not anyway we reporting it, but epididymosomes being present in the aposomes is new to science. And as you could see here, these are the epididymosomes, little higher magnification. These are discrete cylindrical bodies present in the aposomes. They are the epididymosomes. Epididymosomes are already known in literature. Sullivan has done a lot of study on epididymosomes, and he has done assigned a role of positive role of epididymosome contributing to sperm maturation. Over and above the proteins, neurocrine proteins that contribute to sperm maturation, we could find that the apocrine material also has has epididymosomes, and we in further to show that the dense matrix is produced due to amalgamation of these episomes, and the defective spermatozoa get entangled in this dense matrix, and you could see that these spermatozoa are acted upon by material present in the episome or the fused product of the matrix, and you could see that it is formed into total lysis, dissolution of the defective spermatozoa. Now the question is how to connect the epididymosomes of the episomes with what is happening to the spermatozoa. We were lucky, one has got to be lucky to be looking at a good picture that illustrates a hypothesis, a newer finding. What you see here are regions in the episome where you get the epididymosomes, and you have the epididymosomes associating with the defective spermatozoa, and we could see that there are like lipofuscin material produced because of the association of the defective spermatozoa with the epididymosomes of the episome, And so the story is now complete. You have, in those cases, unusual cases of very large, huge number of defective spermatozoa, either due to aberrant spermatogenesis or problem in the post-testicular sperm maturation process. You get defective spermatozoa. They could put a question a survival of the normal sperm. So there is a physiological adaptation whereby the dense matrix of the aposomes containing the epididymosomes, they happen to be produced in large masses. And you remember here, this is how the large extent the aposomes are produced, and these aposomes we have shown to be abounding with the epididymosomes. So we have, you could prove that the epididymosomes have a role via the aposomes to play in the disintegration of the defective spermatozoa. So we could say that in quite a few, starting from the multinucleate giant cell, multiple micronucleate giant cell, and all kinds of problems which we have explained, they were all things were negative happening. But I could want to emphasize here that these are all adaptations. These are all adaptations. Where these are instances where something that is very negative would be projected to be negative. But really, there are mechanisms which are innate and inherent, where the epididymis and the rest of the male reproductive tract has mechanisms which can circumvent such problems and render, even at this stage, produce spermatozoa which are normal. So this came published recently, role of aposomes and epididymosomes in sperm quality control, a light and transmission electron microscopy study in an experimental rat model. Okay, so I'm so fascinated by having been associated with male reproductive biology. Well, I have spent time on many other points, many other issues, many other subjects in male reproductive biology, starting from amphibian reproduction to Sicilian reproduction to, well, but I have carved out a few of my publications, limiting to very high profile focused andrology to explain to you our contribution. It's actually, these were all, of course, under my supervision, my very inquisitive, very efficient, my students, PhD scholars. They were able to spend hours together with the microscope. This is not simply your molecular biology. It is not simply biochemistry, do some small investigations and take data. This is to sit with the microscopes for hours together and go on photographing and then putting them together, assembling them together to tell stories. So I'm so happy that we could spend time with aspects of andrology. And so this is what we did. And I had the fantastic experience of writing a review chapter, the epididymis structure and function, published by my good friend from BHU, Shiv Kumar Singh. The book is Mammalian Endocrinology and Male Reproductive Biology. That was published in 2015, and until now, 2022, we do not have any other publication which could match the reputation, the position of epididymis structure and function that we projected in this chapter, thanks to Shiv Kumar. So I have a lot of course, evidences to project, but I do not want to make tall claims. So finally, to say that male reproductive system is highly vulnerable to toxic infections. The toxicological basis of male infertility is substantiated and explained. Many things which were not known to science. Many of the things which were reported were not known to science. And many of these things which we reported have not been reported again. Some people have adapted our findings to explain what they were making observation. So nevertheless, there are novel mechanisms to circumvent a total collapse, like the microcaniculae, like the Pain-Vaculated epithelial cell, like this dense matrix produced due to disintegrate spermatozoa. They are novel mechanisms to circumvent a total collapse. And that is the reason why there is an adaptation mechanism whereby the infertility, male infertility, is limited to up to like 16%. I hear it for over 30 years. It's not exceeding 16% to the best of my knowledge. And the reason being that parallelly with the reproductive mechanisms succumbing to these toxic manifestations, there is also there are mechanisms which circumvent these issues, and as a result of which, still in these cases, normal spermatozoa are produced, at least through IVF. You will be, we are able to produce the next generation, the young ones, through in vitro fertilization using these spermatozoa. So our observations indicate that male reproductive mechanisms can still offer scope for male contraception. So if we could further concentrate on particularly epididymis as a target for male contraception, but my age is gone. I'm not anymore going to be associated with this study, but the scene is still open. The field is still open, and you can still, somebody youngsters can take up this topic to look at epididymis as target for male contraception. So having said about it, you remember where I started. The first slide, we had a very humble beginning. That was in 1970 with a PhD in 1980. And I said I would reach great heights. I was preemptively saying that I would reach great heights. I'm so happy that my friends from endocrinology and reproductive biology have recognized my contribution to the field of spermatology and andrology. And in this year, that is 2022, has been a really very rewarding year to me. I have been declared one of the 2% top scientists from India on toxicology, 2% of the top scientists in terms of toxicology, and many of my friends have congratulated me. And I have been selected by the Indian Society for the Study of Reproductive Fertility, ISSRO, for the Lifetime Achievement Award. Again, congratulated by some of my friends. So I made a humble beginning. You remember I told you that I started research science in the calotrophic model, reptilian model, and I have gone up to dealing with the human sperm and the variety of animal sperm and animal reproductive biology. I'm so happy. And this study, the whole accomplishment was possible because of people who were supporting me. They were able to bear with me. That's very important. Not only, if they appreciate me or not is a different question. Working with me has been a real tough job. Spending hours together with the microscope. I'm so happy my students really lived to my expectation. Dr. Balasubramanian, he was not my PhD student, he was my student in MSc. Dr. Begain Manivarnan was my student in MSc. Abraham Stanley, starting from there, my PhD student. Well, Dr. Farida Banu, Dr. Sivasamy, Dr. Lakshmi Lada, Dr. Hanayana Avaril, Dr. Kadalmani, Dr. Faisal Kinnotedi, Dr. Agnes Victor Fernandez, Dr. Girija, Dr. Anandi, Dr. Murugayan. Oh, these were all my students at the doctorate level, PhD scholars. And my collaborators, Dr. M. Michael Aruldas from University of Madras and Dr. Ben N.J. Pereira, late Dr. Ben N.J. Pereira from IIT Roorkee. So I thank each one of them for having tolerated me to make this kind of finding. And I thank Department of Science and Technology for the 2 grants, the Indian Council of Medical Research for 2 grants, and University Grants Commission for the 2 grants. And most importantly, these things would not have been possible if I was not thrown with the Christian Medical College and Hospital in Vellore, who were very liberal with me in providing the semi-thin sections and the transmission electron micrographs. And I'm so happy. I could put several of my findings together into one story, from simplicity to a rather complex output. And I'm so happy that I could speak to you. And very especially, I should at this moment thank the colleagues from the AIMS, particularly Dr. Ashutosh Halder and Chaturvedi, and the team of people who they are finding the person to give Dr. K. Laumas Oration, putting the lot on me and throwing a lot on me and giving this wonderful opportunity. Apologizing for the problem in the PowerPoint. I'm so happy that I could make a very good presentation. Thank you so much, and I thank each one of you for your patient hearing and bearing with me. Thank you so much, colleagues. Thank you so much, Director, for this award, and I really miss the thrill of receiving it from your golden hands. But I think I'll get an opportunity to see you sometime as early as possible at your chamber. I'm not, of course, a new person to AIIMS campus. Many times visited, I have dealt with Dr. Farooq, Dr. Rajalakshmi, Asutosh, Dr. Chaturvedi, and many conferences in AIIMS campus. So, but then I still miss the thrill of receiving this award from the director. Thank you so much, sir. Thank you so much.
To conclude the ceremony, I request Dr. Rajiv Kumar, Associate Dean Acad, to deliver the vote of thanks.
Good evening, Professor Akbarsha, the orator for today, Professor Guleria, Director AIIMS, Professor Neera Khanna, Dean AIIMS, Head of the Department, members of the faculty, students and staff, people who've joined in online. Thank you very much for joining us today for this oration. Again, we join in the apology for the delay in starting the program, technical glitches, which none of us are able to handle until they happen at the last minute. Dr. Akbarsha, of course, has had tons of experience and he's been able to put that forward before us. And his passion and his commitment to what he's done is clearly evident in how and what he's presented. As a clinician who deals with these patients on a day-to-day basis, I sincerely hope that one day we will be able to translate the research that he has put forth before us into something that's an actionable target. So I can actually tell my patients we have a therapy for you rather than sending them to the gynecologist for IVF. So we look forward to another oration when we're able to address that point as well. So thank you once again for joining us.